Welcome back to another video. So let's do about antigen recognition by immune cells, recognition units of B and T cells, BCR, TCR, co-repressor, properties of antigen recognized by B and T lymphocytes. And this study note is developed by EPG Pathshala team at Government of India MHRD inspired them. So I would like to thank the developing team of this study material. Thank you so much. I express my sincere gratitude to them. Then antigen recognition by immune cells, recognition units of B and T cells, BCR, TCR, co-repressor, properties of antigen recognized by B and T lymphocytes. So just directly have an introduction about this. Immune system of our body is important and specially designed to recognize and respond to the variety of antigens. Broadly, there are two ways of tackling with the antigen. That is by innate and the adaptive mechanism. Adaptive immune mechanism is initiated. Adaptive immune mechanism is initiated after the effort of innate immune response towards the pathogen is almost over. Adaptive response starts late but lasts for a longer period. The main reason for the late onset is its very precise and species-specific recognition of antigen, unlike that of innate system, where it responds to the general molecular patterns on the bacteria, virus, or other pathogens when they enter the body. The specificity and diversity in adaptive response is attributed to the presence of receptors on B and T lymphocytes as BCR or B cell receptor and TCR or T cell receptor. BCR recognizes the exogenous antigen, but TCR recognizes the endogenous antigen complex with MHC, major histocompatibility complex present on the membrane of all the nucleated cells. The antigen properties thus differ for each of the receptor, and accordingly there are structural variations in their recognition units, which consist of BCR, TCR, co-repressor and other membrane-associated accessory molecules. The recognition unit is a complex of receptors and co-receptors acting as transducer and enhancer to improve the binding ability with the antigen, finally resulting in lymphocyte activation. The different structure of the two types of receptors on lymphocyte and the antigens they recognize is dealt with in this module. Recognition unit of lymphocytes. The B and T lymphocytes are the two specialized cells of the adaptive immunity deputed to recognize and eliminate the intruders of the body in different ways. They vary in the nature of antigen they encounter during surveillance before they mount the immune response, as the antigen can be endogenous or exogenous. The specific recognition of the antigen is the most important function of the adaptive immunity, which is attributed to the membrane receptors on the two lymphocytes. The receptors on the B cells are called B cell receptors or BCR, and those located on the T cells are called T cell receptors or TCR. The BCR recognizes exogenous antigen and forms antibody as the effector molecule after signal from T helper cell through the release of cytokines. The T cytotoxic lymphocyte cell, when recognizes the antigen complex with MHC1, causes cytotoxicity by converting to CTL, but the TH cell complexed with MHC2 present on the membranes of APC, antigen presenting cells, release the cytokines for activation of lymphocytes, macrophage, and inflammatory response. The recognition is followed by response of lymphocytes to the antigen by producing effector molecules, antibody, or effector cells. The information of the receptors is transduced with the help of membrane co-receptors and the co-stimulatory molecule in the cell interior by protein kinase releasing in cell activation. Recognition of antigen, B lymphocyte cytokine release, PCR plus antigen, effector cell, plasma cell, antibody production, cytotoxic T lymphocytes, TC, TCR plus peptide plus MHC1, then cell killing, Effector cell retains specific receptor. Effector cell secretes cytokines. Then cytokine release helper T lymphocyte, TH. Then TCR plus peptide plus MHC2. Macrophage activation, lymphocyte activation and inflammation. Recognition and response of different lymphocytes. Before we understand the receptors of B and T cells, let us know the basic difference of a receptor and a co-repressor. Co-receptor. A receptor is any membrane molecule which receives. Before we understand the receptors of B and T cells, let us know the basic structure of receptor and co-receptor. A receptor is any membrane molecule which receives signals from other molecule with which it binds, specifically called the ligand. Structurally, it has following three components or domains. An extracellular domain that recognizes and binds with the ligand, the transmembrane component that passes through the plasma membrane, a cytoplasmic tail. 4. Transducing the signal from outside the cell to its interior. This is how we represent extracellular domain, plasma. Extracellular domain, plasma membrane, intracellular domain. The signal can be carried to the inside of cell by The signal can be carried to the inside of cell by bringing conformational change in the receptor in the cytoplasmic domain or by taking the help of certain accessory molecules if the receptor is handicapped in signal transduction due to its small cytoplasmic tail. The signal may be by opening of certain ionic channels or activation of certain enzymes like tyrosine kinase, which may trigger important changes in the cell's interior. A co-receptor is also a cell surface receptor and has the ability to bind the signal molecule in addition to a primary receptor for recognition of ligand. It has the ability to bind the signal molecule in addition to a primary receptor for recognition of ligand. Co-receptor is also a cell surface receptor and has the ability to bind the signal molecule in addition to a primary receptor for recognition of ligand. So, a co-receptor is also a cell surface receptor and has the ability to bind the signal molecule in addition to a primary receptor for recognition of ligand. This helps in initiating various biological processes like opening of sodium channels, but in the immune cells, it is for activation of lymphocytes. The immune cells have a number of CD, cluster of differentiation. Co-receptors, which may be monomers or dimers, they are all extracellular receptors. Opening of membrane channels by the attachment of ligands. A co-receptor is also a cell surface receptor and has the ability to bind the signal molecule in addition to primary receptor for recognition of ligand. In addition to ligand. This helps in initiating various biological processes like opening of sodium channels, but in the immune cells, it is for activation of lymphocytes. The immune cells have a number of CD, cluster of differentiation co-receptors, which may be monomers or dimers. They are all extracellular receptors. Okay, you could see a receptor ligand interaction and biological response flowchart. Ligand receptor protein, intermolecular forces, affinity will be there, conformation in receptor protein, biological response. In order to recognize the variety of antigens, which may be either extracellular, exogenous or intracellular, the two types of receptors identify the antigen in a fundamentally different ways. The BCR is located on B cells and TCR is located on the helper T cells, TH cells, and cytotoxic T lymphocytes or killer cell, TCL, the CTL. The BCR or the membrane antibodies MIG recognize the antigen in its native form compared to most of TCR molecules, which recognize the processed antigen located on the antigen presenting cells along with the MHC molecule. T helper cells recognize the antigen in association with MHC2, but the cytotoxic T lymphocytes CTL recognize it and is presented with MHC1 membrane molecule. The TCR T cell receptor also require the co-receptors CD8 plus and CD4 plus on the cells. It is thus important for the BCR to TCR to exhibit molecular diversity to specifically identify the evolving pathogens and variety of newly transformed different cancer cells. Antigen binding site, disulfide bridge, light chain, variable region, constant region, heavy chain, cytoplasm of B cell, antigen presenter of B cell, variable region, constant region, heavy chain, alpha chain, beta chain, cytoplasm of T cell. Structure of BCR complex, MIG and alpha beta heterodimer. The BCR complex has two structurally and functionally different components working as a unit. They are BCR or MIG, membrane antibody, and heterodimer of alpha and beta peptide chains. MIG is for the recognition of antigen and alpha beta heterodimer is for signal transduction. The two functions, when performed separately, give the advantage of using the same cellular machinery for transduction or recognizing a variety of antigens. BCR recognition. BCR recognition, light chain, heavy chain, Ig beta, Ig alpha, ITAM signaling. Then TCR recognition, CD3, then alpha beta, epsilon, delta, CD3, alpha, epsilon, ITAM signaling. This is T cell receptor. This one is B cell receptor, BCR recognition, then light chain, heavy chain, Ig beta, Ig alpha, ITAM signaling. Then TCR recognition, CD3, epsilon and delta, gamma, delta, ITAM, alpha, beta, BCR signaling. Okay, B cell receptor and T cell receptor. BCR or MIG. Membrane antibody has a monomeric Y-shaped structure composed of 4 polypeptide chains and an anchoring unit so as to fix it on the lipid rafts of the plasma membrane along with the other accessory molecules required for signal transduction. Membrane antibody has a monomeric Y-shaped structure composed of 4 polypeptide chains and an anchoring unit so as to fix it on the lipid rafts of the plasma membrane along with the other accessory molecules required for signal transduction. The IgM and IgD are the membrane receptors where the CH4 domain of IgM is hydrophobic to insert into the B cell membrane. Once again, I'm going to read this. Membrane antibody has a monomeric Y-shaped structure composed of 4 polypeptide chains and an anchoring unit so as to fix it on the lipid rafts of the plasma membrane along with the other accessory molecule required for signal transduction. The IgM and IgD are the membrane receptors where the CH4 domain of IgM is hydrophobic to insert into the B cell membrane. The Fab component is oriented towards the cell and Fc is non-covalently involved in signal transduction by interacting with co-receptors and accessory molecules. The cytoplasmic tail of membrane antibody Fc is insufficient to transduce the signal on antigen recognition. Lipid rafts of the plasma membrane provide a platform for selective interaction of the MIG with the signal molecules which carry the information to the cytoplasm for activation of B lymphocytes. Recognition of antigen requires the antibody cross-linking or clustering which initiate the process of B cell activation. The Y-shaped membrane antibody structure has variable light and heavy chains, VL and VH as discussed earlier. So variety of antigens are specifically identified and B cell is activated. Then alpha beta heterodimer. It consists of 2 polypeptide chains alpha and beta which have a long cytoplasmic tail. The cytoplasmic tail of Ig alpha is made of 61 amino acids, but that of alpha Ig beta has 48 amino acids. If we compare the tail length of MIGM with other secretory antibodies, IgA has 14 amino acids, IgG and IgE contain 28 amino acids. The much shorter than the cytoplasmic tail length of alpha beta heterodimer. The cytoplasmic tail of the heterodimer has important component of ITAM, immunoreceptor tyrosine based activation motif. Immunoreceptor tyrosine based activation motif. Immunoreceptor tyrosine based activation motif. Immunoreceptor tyrosine based activation motif, which activate enzyme tyrosine kinase. When an antigen binds tyrosine in these ITAMs become phosphorylated by tyrosine kinase, which are important for transmitting the signal. The efficiency of B cell signal is also increased by the complement activation. The clustering of B cells also brings the complement binding receptors closer, which further attenuates the signal strength. Signal transduction by BCR recognition unit. Here you could see BCR receptor foreign antigen active SYK kinase release signal downstream cytosol plasma membrane active SYK like kinase inactive SYK kinase complement. The sequence of events for recognition and activation of B cell is as follows. Antigen recognition by membrane antibody, BCR antigen clustering, information conveyed to signal molecules on alpha beta heterodimer, activate B cells, ITAM full form, immunoreceptor tyrosine based activation motif. Immunoreceptor tyrosine based activation motif. Activation of cytosine kinase and cell enzymes. Activation of B cell. Activation of B cell. Properties of antigen recognized by the BCR. So, properties, activation of B cell. Properties of antigen recognized by the BCR. The BCR recognizes almost every macromolecule, that is proteins, carbohydrate, lipids, etc. that can bring the clustering of membrane antibody. The BCR identifies only a particular area of the macromolecule of protein antigen called the antigenic determinant or epitope. When the macromolecule exhibits many antigenic determinants, then it is called a multivalent or polyvalent antigen. A B cell may recognize the antigen with epitopes which may be identical or non-identical, identical in chemical structure. The BCR identifies only a particular area of the macromolecule of protein antigen called antigenic determinant or epitope. When the macromolecule exhibits many antigenic determinants, then it is called a multivalent or polyvalent antigen. The B cell may recognize the antigen with epitopes which may be identical or non-identical, identical in chemical structure, repeating or non-repeating. Cellular spaced identical epitopes are different, overlapping or non-overlapping. Overlapping if epitopes are closely placed. Conformational or non-conformational, linear or three-dimensional epitopes. Since the pathogen display variety of multivalent antigens, either proteins or carbohydrates, they induce the fast signal transduction across the B cell membrane due to clustering of BCR. The overlapping epitopes can result in antibody clustering better than the non-overlapping. The protein antigens may be linear or they assume three-dimensional structure. The denaturation of protein may result in disappearance of certain antigenic determinants which are called the conformational epitopes. There are four types of monovalent non-covalent interactions involved in antigen and antibody interactions, which are ionic, hydrogen hydrophobic, and van der Waals forces. It should be very clear that not all the antigens which are recognized and bind with the receptor on lymphocytes are capable of triggering their activation. These molecules which are activating the lymphocytes are called immunogens. Activating lymphocytes are called immunogens. These are small chemicals which may be successful in binding to the membrane antibody but do not activate the lymphocytes. So these chemicals like dinitrophenol are called haptens. These chemicals like dinitrophenol, they need to help of some carrier protein, making a protein carrier conjugate which converts them to immunogen. Hapten do not activate B cell BSA, bovine serum albumin, is an example of hapten. Then carrier protein BSA, bovine serum albumin, carrier protein BSA, activated B cells. So just bovine serum albumin is a hapten, but when it combines with the carrier molecule, it acts as an immunogen which creates activated B cells. Like activating lymphocyte. Structure of TCR recognition unit. T cells recognize the antigen by the T cell receptor or TCR. T cell recognize their antigens by the T cell receptors or TCR. Each T cell has approximately 50,000 to 100,000 identical receptors for antigen. They are clonally distributed, which means each clone has one type of TCR. TCR is membrane bound only and is not secreted like BCR. It also has a 3-domain structure comprising of extra cytoplasmic part, transmembrane region, and cytoplasmic tail as discussed earlier in for BCR. Alpha chain, beta chain, variable region, constant region, transmembrane region. A complete recognition unit. A complete recognition unit of T cell or TCR complex consists of the following important components: 2 alpha and 2 beta or 2 gamma and delta peptide chains expressed on surface of the T lymphocyte as TCR. CD3 complex or invariant chain consisting of alpha, delta, eta and epsilon. Co-receptor molecule which is either CD4 plus or CD8 plus. There are two distinct subtypes of T lymphocyte based on TCR composition, that is the TCR with alpha and beta or gamma and delta peptide chains. There is a difference in recognition of antigen as the alpha beta T lymphocyte recognizes the antigen presented on MHC-LA molecule, which is a polymorphic form unlike that of MHC-IB, which is non-polymorphic, and the antigen complex with MHC-IB is recognized by gamma delta TCR. Gamma delta TCR have affinity for the non-protein antigen, which may not be complexed with MHC molecule. It recognizes the conserved structures on the invading microorganism. TCR is a highly variable heterodimer. TCR is a highly variable heterodimer and is structurally homologous to Ig superfamily. The heterodimer has two functionally different domains, that is variable and constant. The variable domain is responsible for the antigen recognition and binding, but the constant domain is transmembrane with a small cytoplasmic tail. The transmembrane domain has a stretch of positively charged amino acids which allow them to interact with the CD3 molecules. CD3 complex chains are called invariant as they have no variability in the amino acid sequence. It has three heterodimers of alpha, delta, alpha, epsilon, alpha, delta, and homodimer chains of heterodimer. Homodimer of zeta. Yeah, that letter is zeta. Zeta. Homodimer of zeta is more prevalent on the T cell surface than the heterodimer peptide. The gamma, delta, and eta chains belong to immunoglobulin superfamily, but zeta chain is different and has a very long cytoplasmic tail consist of about 113 amino acids. The cytoplasmic tail of CD3 is associated with the special peptide motifs called ITAM or immunoreceptor, which are tyrosine-based activation motifs required for the activation signal of the T cell. There are 3 ITAMs on zeta and 1 on each on delta, eta, and gamma chain CD3 molecule. The TCR triggering also undergoes receptor clustering and conformation change in CD3 molecule along with the receptors and MHC molecule. MHC2, AG, TCR, CD3, CD8. Antigen and the recognition unit of T cells. Antigen receptors, killer cells, CD8 protein, T cell receptor, antigenic peptide, MHC class 1 cell membrane, infected cell. Then helper T cell, CD4 protein, T cell receptor, Antigenic peptide class 1 MHC, class 2 MHC, cell membrane antigen presenting cells. The CD4 and CD8 co-repressors act as enhancers which increase the affinity of TCR with MHC1 or MHC2 molecule. CD4 is the co-receptor for Th cells which binds with the MHC2 of APC, whereas CD8 bind with MHC1 exhibited on all the nucleated cells. T lymphocytes with CD8 surface glycoprotein are called CD8 plus T cells, and those with CD4 are called CD4 plus T cells. CD4 and CD8 have Ig fold domain like structure, where CD4 is a 55 kilodalton glycoprotein expressed on approximately 5 to 6% of T cells in human. It is a single peptide alpha monomer which is linked to the IECK type of tyrosine kinase on the cell membrane. It consists of 4 extracellular domains in peptide gene. It is the important receptor for the entry of HIV, human immunodeficiency virus, which cause deadly disease AIDS, acquired immunodeficiency syndrome. The cells with CD4 glycoprotein are more prone to attack by HIV. So CD4 count, which is a lab test done on AIDS victims, indicate the health of patient. It is possible to perform a CD4 co-receptor blockade using antibodies in order to lower T cell activation and counteract various disorders leading to immunodeficiency and autoimmunity. It is possible to perform a CD4 co-receptor blockade using antibodies in order to lower T cell activation and counteract various disorders leading to immunodeficiency and autoimmunity. So CD8 is a 34 kilodalton alpha beta heterodimer, both with extracellular transmembrane and cytoplasmic domains. It is present on 30% of T cells in blood of human. It consists of only one extracellular domain of the peptide and is associated with ICK tyrosine kinase in the cytoplasm. The extracellular domain of CD8 alpha interacts with alpha 3 domain of class 1 MHC molecules, which help in increasing its sensitivity and stabilizing the TCR and MHC interface required for the cell activation. CD4 N-terminal. CD4 N-terminal AH-VH region, CD8 C-terminal, CD8 N-terminal, N-terminal, C-terminal, C-terminal. Receptors co-repressors CD4 and CD8. CD4 N-terminal, C-terminal. CD8 N-terminal, alpha, beta, N-terminal, C-terminal, C-terminal. The recognition of antigen by T lymphocyte results in signal transduction via reversible phosphorylation of tyrosine by protein kinase and phosphatase. T lymphocytes have 5N SRC family of kinase and signaling to phosphorylate ITAM located on CD3 and zeta genes. The signal step and TCR triggering involves different kinase like LCK, FIN, ZAP70 and tyrosine phosphatase CD45. It is membrane protein which has tyrosine phosphatase to activate SRC family kinase. The regulatory mechanism for weakening or attenuation of the immune receptor signaling is important to prevent reactions for self-antigens. It is by the inhibitory receptors located on B and T cells which have ITIM immune tyrosine inhibitory motors to activate phosphatase. Properties of antigen recognized by T cell. The antigen recognized by T cells are small linear peptides loaded on the MHC1 or MHC2. They identify protein antigens which are processed and thus broken into small peptides. The reason only proteins are recognized is due to the peptide compatible structure of MHC. MHC molecules show polymorphism and have the ability to bind with a variety of peptides. Out of the large repertoire of MHC molecule, T cell recognize peptides which is complex with one of the MHC molecule, and it is called MHC restriction. T helper cells with CD4 plus and are class 2 MHC restricted, whereas T cytotoxic cells with CD8 plus are MHC1 restricted. The polymorphic domain of amino acid residues of MHC are near the peptide binding cleft, whereas The non-polymorphic Ig-like domains of MHC molecule has the binding site for either of the two subpopulations of T cells, that is Th or Tc. The class II restricted T helper cells, Th cells, are activated by the extracellular antigens which pass through the endosomal vesicles for antigen processing, unlike that of class I restricted cytotoxic C cells which bind with the intracellular antigens produced by viral infections or cancer proteins after passing through cytosolic processing. As it is difficult for the T helper and T cytotoxic cells to distinguish the endogenous or exogenous antigens, they are directed by the two types of MHC molecules, that is MHC1 and MHC2. The T cells recognize not all the peptides which are processed, but a few of them which are more compatible with the MHC, and this is called immune dominance. So gamma delta T cells are not MHC restricted as they recognize the antigen without MHC molecules also. There is a small population of natural killer T cells which have the characteristic markers of NK and T cells, and they recognize non-protein antigens which are not MHC restricted. The recognition of antigen by the two different lymphocytes is different mechanism. B cell recognition of epitopes of antigen has following features. The interaction of B cell with epitopes form binary complex, that is AG-AB. B cells bind with soluble antigens. B cells do not require MHC molecules for epitope recognition. B cells recognize antigen which may be proteins, polysaccharide and lipids in nature. B cells recognize the epitopes which are accessible hydrophilic mobile peptides with sequential and non-sequential amino acids. T cell recognition of epitope of antigens show following features. The interaction with the antigen and T cell receptor forms a ternary complex, that is AG-MHC-AB. The T cell do not bind the soluble antigen. The T cell require MHC molecule for the presentation of processed antigen. The T cell recognize the antigens which are mostly protein in nature. The epitope recognized by T cells are linear peptides due to processing of antigens and are bound to MHC molecule. Next, quick summary. The receptor is a membrane bound. The receptor is a membrane glycoprotein consist of extracytoplasmic transmembrane and cytoplasmic tail. Adaptive immune response is specific because of receptors on B and T lymphocytes called BCR and TCR. They work in coordination with co-receptors and accessory molecules, thus forming BCR and TCR recognition units or complexes. The BCR unit or complex consists of membrane antibody, MIG, and alpha beta heterodimer as to receptors. The alpha beta heterodimer have ITAM for signal transduction. Membrane antibody clustering is important for antigen recognition by BCR and cell activation. ITAM triggers the signals to the cell interior by activation of protein kinase. To T lymphocytes recognize the antigen complex to the MHC1 or MHC2 molecules associated with co-receptor CD4 and CD8 invariant chains of CD3. T helper cells recognize antigen complex with MHC1, MHC2 on APC and co-receptor CD4. T cytotoxic cells recognize the antigen complex with MHCl and co-receptor CD8. CD4 is a monomeric glycoprotein, but CD8 is heterodimeric. CD4 interacts with alpha-3 domain of MHC8. There are 3 ITAMs and on zeta and 1 on delta, eta and gamma chains of CD3 molecule. TC and TH cells are MHC restricted. The epitope properties of antigen recognized by T cell are different from T cells.